So in DNA replication, it is synthesized from the 5' side to the 3' side of the strands. The first step is helicase unwinding the parent DNA strand by breaking the hydrogen bonds between nucleotides of the parent strand, which will cause the DNA to go from double stranded to single stranded in two sets, the leading strand and the lagging strand. The second step is the topoisomerase maintaining the fork structure of the and loosening the strand. Third step is that now that the strands are loosened, RNA primers serves as a starter sequence for DNA polymerase, and then DNA polymerase 1 can now start to synthesize the new strand by adding nucleotides together to the new strand after removing the RNA primers. The fourth is DNA polymerase 3 starting at the leading strand, which it first replicates the DNA off of, which is then copied to the lagging strand. Then ligase will bind these fragments from the template strand via forming phosphodiester bonds to the DNA backbone from the lagging strand.
Showing you how transcription works in the protein synthesis process with cards. So the top row here, I have represented the 5' to 3' direction, and the bottom row should be the 3' to 5' direction. So as for the cards themselves, I'll be using numbers and royal cards. So that includes Jack, Queen, and King. So the numbers, they represent thymine, uracil, and adenine, while the royal cards represent um, cytosine and guanine. Specifically, the the red would represent the thymine slash uracil. The black would represent the adenine. The red royals would represent the cytosine, while the black royals would represent the guanine. Um, okay, so before transcription begins, you have this RNA polymerase, right, which is uh like it makes the mRNA. So before it even starts, it attaches to what's called a promoter region, which is a series of thymines and adenines that tell where the that show the RNA polymerase where to start making mRNA. So once the RNA polymerase attaches to the promoter region, the actual transcription process can begin. So uh, to transcribe this DNA will go from the 5' to 3' direction. So we'll be using this bottom strand as a template since it runs from 3' to 5'. So here I'll just move these. We have a red number, so that would represent a thymine. The corresponding nucleotide would be adenine, so we can just put an adenine right here. Uh, we have well, we have two black royals that would represent guanine. So we can put two red royals, which would represent the two cytosines. Um, we have another red number, which would represent um, thymine. So with thymine, we add an adenine, and we have a red royal, and that would represent um, thymine. No, sorry, it would represent cytosine, and cytosine would go with guanine, so we add a guanine. And this last one, it's slightly different because it's adenine, which is represented by a black number. So instead of thymine, we would add a uracil, which is what separates DNA from RNA, the fact that uracil is there. And that should be it for our transcription process. So in order for the RNA polymerase to stop, it has to reach a terminator region, which basically tells it like when it can stop coding for a gene or a protein. Yeah, that is transcription using guards. Thank you.
Before this pre-mRNA molecule can become a full RNA molecule, it has to go through several RNA editing steps. The first of these is the addition of the GCAP on the 5' end, which helps protect the mRNA from degradation by enzymes and helps the ribosomes attach to the 5' end in the cytoplasm to start translation. The second step is the addition of the poly A tail on the 3' end, which helps transport the mRNA out of the nucleus. And then finally, the last step is RNA splicing, where spliceosomes take out the introns and connect all of the exons together. And this would be a full RNA molecule. Can also go through a process called alternative RNA splicing, where the gene can create different polypeptides depending on which segments are treated as exons during the processing stage. So normally, the spliceosomes would remove each of these introns and combine the 4 exons, but if it removes the introns, and then also removes exon number 4 and treats it like an intron, then these 3 exons would create a completely different polypeptide.
Translation is a process that occurs in the ribosome, and it involves the formation of polypeptide chains. And this starts with a strand of mRNA that has already been edited and is ready to be translated. So the first step of translation that actually happens is that a small ribosomal subunit will bind to the strand of mRNA, and then a initiator tRNA, which is transfer RNA, will bind to the 5' cap of the mRNA strand and scan it until it finds the start codon, which is AUG. The binding of the initiator tRNA to the edited strand of mRNA will signal for the second part of the ribosomal subunit, the large subunit, to enter the picture. And what happens is that, just give it a second, the initiator tRNA will enter the P site of the large RNA subunit. What the P site is it is the part of the large RNA, the large ribosome subunit, which actively holds the polypeptide chain. There are 2 other sites. The A site holds the polypeptide or the amino acid, which is going to be incoming, and the E site is for tRNAs that no longer have amino acids attached to them to exit. Once the initial tRNA has been bonded and the chain has begun, the ribosome will move down the strand of mRNA and see the next polypeptide, the next amino acid it needs to add to the chain. And so this mRNA codon says UAU, which means that it codes for tyrosine. So tyrosine will be taken. A tRNA will go through the cytoplasm until it finds an amino acid of tyrosine, and then it will bring that amino acid to the ribosome where the polypeptide is being assembled and take it to the A site. Okay. Once the tyrosine molecule has had its anti-codon binded to the mRNA's codon and it has reached the A site, before transitioning, the tyrosine will form a covalent bond with the carboxyl group of the methionine. So prior to the the tRNA carrying the tyrosine entering the P site, it'll form a bond. It'll form the covalent bond with the methionine, and then once that covalent bond is formed, the tRNA carrying the methionine will move to the E site, and the tRNA carrying the tyrosine. One second. That will move to the P site. This process is called translocation, and essentially what it means is that, and it expends in order to get the energy to do this, you need to break apart one GTP molecule, which is guanosine triphosphate, and so the energy from that will allow you to move the tRNA that was carrying the methionine into the E site, where from this point it can easily exit. And now the tyrosine is in the P site, and it is the tRNA that is actually carrying our amino acid chain. Okay, at this point, you will see that now that we have bonded the methionine and the tyrosine, we will move down the mRNA strand, and then this UGG is a stop codon. So what this codes for is that a protein called a release factor will actually come from the cytoplasm and into the A site. And the release factor will signal the large subunit to initiate hydrolysis between the tRNA in the P site and the polypeptide chain. So it'll signify hydrolysis, and it'll break apart the polypeptide chain, which is now done. It's completed. And then once that is done, the release factor will signify to this entire translation complex that it needs to essentially just break up. And that also requires two guanine triphosphate molecules. So you break off one end of the phosphate, and then before that though, this tRNA will exit because it can still be used later on to bring another tyrosine amino acid for another polypeptide chain. But right now, the ribosomal site will break up, and that is signified by the release factor. It indicates that, and that requires, like I said, 2 guanine triphosphate. So all of these will disperse. The large subunit, everything, this entire complex will disperse. It's not actually going to break like this, but these parts may be reassembled for later, or they may be destroyed in lysosomes. And we have our end product, which is our amino acid, which can be used for whatever the cell sees fit, and that is translation.
Gene expression is how DNA and proteins are controlled in cells to express certain genes. All cells in the human body have the same DNA, but different cells turn on and turn off certain genes. Genes can be controlled in all aspects of transcription and translation processes. DNA is tightly wrapped around proteins called histones. Tightly wrapped DNA tend to be turned off since it's harder to access for RNA polymerase. Acetylation of histones enable transcription, turn on genes, attachment of COCH3 or the acetyl group, while methylation of DNA blocks transcription factors, attaches CH3 or the methyl group. For RNA synthesis to start, there needs to be binding between the promoter region, RNA polymerase, and the enhancer region to form a transcription initiation complex. Silencer proteins can bind to the enhancer region to block gene transcription. siRNA can bind to the mRNA as death tag because it creates a double-stranded mRNA. In post-translation, ubiquitin can bind to proteins to degrade and cycle in the proteasomes.